In this video, we are going to be covering the topics in Chapter 20, which has to do with the electron transport chain and oxidative phosphorylation. This figure is showing you a partial picture of the electron transport chain. So as you can see here, this specifically is looking at the mitochondria. Okay, so remember, we already talked about the different components of the mitochondria itself. So this lipid bilayer that we have is the inner membrane of the mitochondria. The intermembrane space is going to be the space that it is present in between the inner membrane and the outer membrane of the mitochondria. The matrix is the space inside the inner membrane of the mitochondria. So these complexes that we're going to be transporting electrons in the system, understand that they are embedded in the inner membrane of the mitochondria. This is just illustrating 3 out of the 4 complexes that is there. This is just trying to give you an overall view of what is happening in this situation, but we will be looking at these processes in detail in the following slides. So understand that overall, the role of the electron transport in metabolism is specifically how the molecule of oxygen is going to be accepting electrons in order to form water. Remember that we already discussed that in the process of respiration, we're going to take glucose and we are going to be converted into CO2, water, and energy. Okay? We already saw in Chapter 19 that the CO2 is specifically formed in the TCA cycle. We're going to see in Chapter 20 how the water is made at the end of these electrons that are transported. In the energy, we have so far seen that we have made in terms of energy being released, we have seen which process end are exergonic or endergonic. But here, when it comes to energy, I'm going to talk about the ATP synthesis. So far, we have seen that we have made 2 ATPs in the process of glycolysis, and we have made 2 ATPs in the TCA cycle. So overall, as I was mentioning, remember, oxygen is going to be accepting these electrons and converting it, and oxygen itself is going to be converted into water. Now, one of the reactions that we are going to be observing here is how these coenzymes that we were talking about and were synthesized in glycolysis and also in the TCA cycle are going to be converted ultimately to ATP. The reaction that is going to be occurring and we are going to be observing, it is called oxidative phosphorylation. So you may be wondering, why is it called oxidative phosphorylation? It's because those 2 reactions are going to be occurring in the process. This is known as oxidative phosphorylation because, as you can see here, the NAD and FADH2 is not expressed in these equations, but understand that a similar reaction is going to happen. So NAD is going to be oxidized to make NAD+. as you can see, that results in the generation of electrons. Those electrons are ultimately going to be accepted by oxygen to make water. That's why the overall reaction of the oxidation of NADH to NAD results in oxygen being the electron acceptor, meaning in this case it's going to get reduced to make water. Now, phosphorylation, we are going to see in detail later how a proton gradient is going to be created, and ultimately a complex that independently is called ATP synthase is going to be generating ATP by combining ADP and phosphate. So understand that the process of oxidation and phosphorylation are coupled to one another by a proton gradient, which is a term that I introduced in the previous slide. So overall, understand that what is occurring is that in this process, when these molecules of NADH are getting oxidized to NAD+, or as we're going to also see, FADH2 gets converted into FAD, one of the things that happens is that we are going to have protons, as you can see here, move into the intermembrane space. So that's why these arrows in purple are illustrating. Okay, when that occurs, understand that then we're going to have a high concentration of H plus in the inner membrane space of the mitochondria, which means that it's going to be low pH. As a result, we are going to have a high pH in the matrix of the mitochondria because we will have a low concentration of H plus. Understand that these electrons that are going to be transported is what leads to the creation of the proton gradient. And we are going to see that in detail when we talk about each of the reactions that is happening in the complexes that are involved in the electron transport chain. So now we're going to look in detail at the organization of the transport complexes or the electron transport complexes inside of the mitochondria. So there are 4 major complexes. So even though in the figure in this slide you see 5 complexes, complex 5, which we're going to treat as a separate entity, this is ATP synthase. And you're going to see here how overall this complex 5 is going to be creating the ability of those protons to come back into the matrix in order to balance out that proton gradient that was created, and then that's how the ATP is synthesized along with the water molecule once oxygen accepts electrons. Okay, so understand that the electrons are going to move from low to high reduction potential, but we are not going to talk about the chemistries of the reduction potential. And ultimately, remember, oxygen is going to be accepting those electrons in order to make water. Let's now look at the molecules that are involved in transporting the electrons specifically. So, understand that we are going to be observing cytochromes and non-heme iron proteins doing the transporting of the electrons in this system. Okay. One that is not illustrated here is coenzyme Q or ubiquinone. But understand that cytochromes, non-heme iron proteins, and ubiquinone are the ones that are going to be doing the transporting in the electrons. It's just that physically, the cytochrome and the non-heme iron proteins, we can actually have the electrons being accepted in them. And the reason is because when we look at these molecules, as you can see, they can have an iron 3 plus accept an electron and they make iron 2. So a cytochrome is the molecule that we have on the left. The non-heme iron proteins are the ones on the right. Now, you guys don't need to memorize these structures. You don't need to draw them out. You don't need to submit the structures. I just wanted to illustrate to you guys how complex inorganic molecules are also present in such small compartments of the cell. Now let's go into detail through each of the complexes and what is their involvement when it comes to transporting electrons in the electron transport chain. Starting at complex 1. Complex 1, it is known as NADH CoQ oxidoreductase. Okay? So in NADH CoQ oxidoreductase, what we have is that NADH is going to be converted into NAD, and as you can see here, we're going to have flavin mononucleotide will be accepting those two electrons and is going to have its reduced form. Then the reduced form of that flavin mononucleotide then is going to transfer electrons to the non-heme iron protein, which ultimately those electrons are going to be accepted by Q, and Q is ubiquinone, and ubiquinone is going to be then changed into ubiquinol, which is the reduced form of ubiquinone. Okay, so let me just write down QH2 is the reduced form of ubiquinol. Okay, and then so far as you can see, ubiquinol is then going to be transporting these electrons to further complexes, as we're going to see in later slides. Okay, so as you can see, the path for electrons goes NADH, okay, then they get passed on to flavin mononucleotide. Then we have the non-heme iron protein, and then we have CoQ, and then CoQ gets converted into CoQ, and Q is the same. So let me just write it here. Then this is QH2. Understand that during this process, there are also going to be protons pumped. Specifically, 4 protons, and I'm going to highlight that. 4 protons from the matrix of the mitochondria are going to be pumped into the inner membrane space of the mitochondria. Let's move on to complex 2. So complex 2 is called succinate CoQ oxidoreductase. So understand that this is specifically one of the steps that is involved in the TCA cycle. So the enzyme succinate CoQ oxidoreductase, which is directly involved in electron transport, is the enzyme that takes succinate to fumarate in the TCA cycle. Remember that in that process, we actually took FADH and we converted it to FADH2. So as you can see, FAD got into FADH2 by reduction, but understand that this FADH2 then is going to produce 2 protons into the matrix, but is actually going to release some electrons that are accepted by the non-heme protein. And then we are also going to have ubiquinone be converted to ubiquinol. Okay, so understand that ultimately here we have the transfer of the electrons from FADH2 to coenzyme Q, which is going to make QH2, and that's how the electrons will go from either complex 1 and complex 2, and they're going to be landing on the next complex, which is complex 3. Complex 3 is known as cytochrome reductase, okay? So understand that a series of cytochromes in iron sulfur proteins are going to be involved in this process. I'm not going to be talking about those reactions in detail. What I mainly want to remind you is that complex 1 pumped protons, complex 2 didn't have any pump of, it was not pumping protons from matrix to inner membrane space of the mitochondria. Okay? Understand that here specifically, at this step, what happens overall, okay, is that we are going to have then this cytochrome, or sorry, this ubiquinol molecule is going to be dumping these electrons into complex 3, and basically at the end of all the series of reactions that is occurring, okay, because remember, this is the ubiquinol that came from complex 1 and complex 2. After all the reactions happen, which as you can see, there's a whole bunch of electrons happening, overall cytochrome C is the one that is going to be transporting, is going to be the new vessel for transporting these electrons into the next complex, okay? Understand that specifically, once we are on complex 3, protons are going to be pumped again from the matrix of the mitochondria into the inner membrane space of the mitochondria, okay? So understand that it doesn't matter if ubiquinol came from complex 2 or it came from complex 1. At the end of the day, 4 protons will be pumped, okay, by NADH2 or NADH, because remember that the ubiquinol came from those 2 complexes. So in complex 3, we have an additional 4 protons that are pumped. Lastly, these electrons that were transported from cytochrome C land on complex 4. On complex 4, then those electrons are going to make all the way down. As you can see here, the electrons are coming down. They're going to be accepted by some systems, but ultimately, the ultimate acceptor, okay, of this process is going to be oxygen. Oxygen is going to be accepting these electrons, is going to be combining with protons, and we are going to be making water, okay? So ultimately, on complex 4, which is called cytochrome C oxidase, we are going to be having oxygen reduced to water. Understand that complex 4 also pumps 2 protons, okay? So far we have seen that complex 1, 3, and 4 are proton pumps. And that's what leads to the formation of that proton gradient that I explained previously in this video.